Good afternoon. Thank you for the opportunity to present my work. Today I will talk about thymus hypoplasia resulting from 22q deletion syndrome. 22q deletion syndrome is one of the most common chromosomal microdeletion. It affects 1 in every 4,000 births. Children born with this syndrome have 1.5 to 3 megabase deletion on chromosome 22. These children are born with defects in multiple organs. It has been shown that most of the congenital malformations in these children are due to haploinsufficiency of gene TBX1, which codes for a developmental transcription factor. One of the common malformations due to this syndrome is thymus hypoplasia, which occurs in 60 to 70% of the patient. This results in T-cell lymphopenia of varying severity. Most of the patients develop normal T-cell subset but reduced T-cell numbers. Less than 1% of the patients are born without thymus and they need allogenic thymus transplant for their survival. Some other congenital malformation these children suffer with are hypoparathyroidism, second-degree heart field defects, and dysmorphic facial features. We are interested to know what causes thymus hypoplasia in 22q deletion patients and can this be corrected with the bioengineering techniques. Most of the congenital anomalies associated with this syndrome, such as thymus hypoplasia, hypoparathyroidism, and cardiac defects, all are linked to developmental problems of the pharyngeal region. In mice, the third pair of these pharyngeal pouches, PP3, each forms a single epithelial organ primordium surrounded by a mesenchymal capsule, which later develops into one thymus lobe, which is red in color, and one parathyroid gland, which is blue in color. TBX1 requirement has been established for the development of all the pharyngeal structures. TBX1 is essential for the early thymus development, but it has not yet been linked directly to thymus fate. As we see, a sagittal section of E10.5 and TBX1 is expressed in the core mesenchyme of the pharyngeal arches. TBX1 is required for the pouch formation and patterning. We are keen to know how TBX1 haploinsufficiency in this syndrome results in the thymus hypoplasia. To study these, we have used TBX1 Neo2 mouse models, which are kind gift from Dr. Balini. These mice are designed in such a way that mice with homozygous allele for TBX1 Neo2 expresses only 34% of TBX1. TBX1 Neo2 homozygous mice are neonatal lethal and die within a day or two post-birth. We are seeing the live cell image of cardiothoracic region in a E13.5 wild type and E13.5 TBX1 Neo2 mutant embryo. Thymic lobes are demarcated in black dotted lines. We see normal lobes are bigger and oblong in shape, while hypoplastic lobes are smaller and irregular in shape. These lobes were then sectioned transversely and stained for the stromal cell population. Red and purple color stained for mesenchymal cells, which form the thymic capsule and subcapsule, while green color stained for the epithelial cells, which are required for the T cell development. Except being smaller in size, the hypoplastic lobe structurally appeared similar to normal lobe. Not only the structure, but hypoplastic lobes have similar composition as normal lobes. Normal and hypoplastic embryonic thymus both have similar percentage of mesenchymal cells, thymic epithelial cells, and early thymic progenitors. The overall cellularity is reduced due to the small size of the hypoplastic lobe. The mesenchymal cells and the epithelial cells also are in reduced number. However, despite the lower number of the stromal cells, the percentage of the stromal cell is similar in normal and hypoplastic lobe. This left us to think that It's not the percentage of the stromal cells, but the functional problem in the stromal cells which is preventing the expansion of the hypoplastic lobe. This could be due to the defect in mesenchymal population or epithelial population or both. Identify which stromal cell population is functionally compromised in 22q deletion thymuses, we did fetal thymic organ culture. In this technique, paired thymic lobes from E13.5 gestational age embryos are plucked and placed over the membrane. This membrane is resting on the sponge which is submerged in the growth media. This is allowed to grow for 8 days. After 8 days, we see a nice grown expanded lobe which can support thymopoiesis. Similarly, hypoplastic lobes were plucked and placed over the membrane. For the hypoplastic lobes, we see different phenotype. For the first phenotype, we see that the lobe has completely disintegrated and we see nothing but cell debris. This could not support thymopoiesis either. In the second phenotype, we see a small tissue has formed. This has a better survivability and can support thymopoiesis to some extent. In the third phenotype, a small lobe has formed which can support thymopoiesis as good as normal lobe, but still it has 3 times less cellularity than the normal lobe. About 60% of the times we see hypoplastic type 3 phenotype, which we have termed as small lobes, and 40% of the times we see type 1 and type 2 lobes. This perfectly corroborates with the patient data where we see varying degree of T-cell lymphopenia. A little modification in the previous technique can help us to identify which of the two stromal cell population is functionally compromised in the hypoplastic lobes. E13.5 thymic lobes were isolated, and before placing them on the membrane, they were first disintegrated to single cells. These cells were then sorted to different populations. The blue dots are the mesenchymal cells, the yellow dots are the epithelial cells, and the purple dots are double negative thymocyte, fibroblast, and other population. These different sorted populations were then mixed back together and overlaid on the membrane and allowed to grow for 10 days. If our hypothesis is correct, then replacing the stromal cell population of hypoplastic lobe with normal stromal cell population should be able to restore the growth of the hypoplastic lobe. We individually check two cell population, mesenchymal cells and epithelial cell. In the mesenchymal substitution set, we replace the hypoplastic mesenchymal cell with normal one. And in the epithelial substitution set, we replace the hypoplastic epithelial cell with the normal one. We see that when a normal lobe disintegrated and reaggregated back, it formed a big lobe-like structure which can support the thymopoiesis. When a hypoplastic lobe was disintegrated, it could not reaggregate back, neither could it support thymopoiesis. In the mesenchymal substitution set, where we have replaced the hypoplastic mesenchymal cells with normal mesenchymal cells, could form a nice big lobe and support thymopoiesis as good as the normal reaggregated lobe. In a epithelial substitution set where we have replaced the hypoplastic epithelial cells with normal epithelial cells, this could not restore the thymic lobe, neither could support thymopoiesis. We did this experiment multiple times and we see that every time mesenchymal cell replacement could effectively restore the cellularity of hypoplastic lobe. But epithelial cell replacement could never do so. So how do mesenchymal cells support thymus tissue development? It has been discussed earlier that growth factors, cell adhesion molecules, matrix proteins, and cell-cell contact play important role in proper expansion of thymus tissue. To see what's lacking in hypoplastic lobe, we performed 10x single-cell RNA sequencing to query all the cell population. Single-cell RNA sequencing revealed 17 clusters in mouse embryonic thymus of gestational age E13 to 13.5. We see one endothelial cluster and 6 epithelial clusters labeled as E1, E2, E3, E4, E5, and E6. We see 5 mesenchymal clusters labeled as M1 to M5, and we see 4 hematopoietic clusters labeled as H1 to H4. Same number of clusters were observed in the 22q hypoplastic lobe, but we see overexpression of cluster M5, E5 and underexpression of cluster E6. We did single-cell RNA sequencing for another mutant which causes thymic hypoplasia due to mutation in transcription factor FOXN1. We see similar clusters here, but different pattern of over and under expression of clusters. Gene expression analysis revealed that M5 cluster, which is highly overexpressed in TBX1 Neo2 mutant, is composed of vascular smooth muscle cells, while E5 and E6 belong to thyroid and parathyroid epithelial cells. Some of the genes which are overexpressed in cluster M5 are actin-1, CAP-N6, elastin, and amylin. We see many extracellular matrix protein genes which are overexpressed in TBX1 Neo2 mutant mesenchymal cells. We propose that the overexpression of these ECM proteins probably hinders the expansion of the hypoplastic lobes. Mesenchymal colony assay has also revealed that mesenchymal cells from hypoplastic lobes have attenuated survivability. We isolated the mesenchymal cells from normal and hypoplastic lobes and seeded equal number of cells in 6-well plate supplemented with special media. After 15 days, we see that hypoplastic mesenchymal cells have formed colonies which are much reduced in size compared to the normal mesenchymal cells. These experiments are still under progress, and with this I would like to conclude my talk. We could identify that thymus hypoplasia due to 22q deletion syndrome is linked to mesenchymal cells defect. 10x single-cell RNA sequencing has also revealed mesenchymal transcriptome is heavily impacted in 22q deletion. Similar has been suggested by mesenchymal colony assay. In future, we would like to do some regeneration assays with normal mesenchymal cells to enable thymus organoid formation in clinical settings. I would like to acknowledge Dr. Nikolai Van Oyers for giving me the opportunity to work on this syndrome, my lab members for their continuous help and support, all the core facilities and the collaborators, and I thank you all for being attentive during my talk. With this, I would like to take the questions. Thank you.